Good morning, good afternoon, everyone. I'm very happy to be presenting our data on the gut microbiota and vaccine responses in infants exposed to HIV. I think everybody knows in this room that the infant gut microbiota is very important in determining the infant's immune and metabolic trajectory. And the most important determinant of an infant's gut microbiota is his or her maternal microbiome. And there's some evidence that this early life gut microbiota in infants may be associated with differences in vaccine immunogenicity. For example, bifidobacterial abundance and antibody responses in infants seem to be associated. So we study BCG vaccine because it's a live attenuated mycobacterial vaccine that's the prototypic neonatal vaccine. It elicits robust Th1 CD4 T cell responses in neonates who are notorious for having a Th2 skewed immune response. It protects against pulmonary and disseminated TB in children, but with variable efficacy. There's multiple reasons that probably contribute to this variable efficacy, but microbiota might be one of them. HIV-exposed infants are infants that are born to mothers with HIV, but who are uninfected themselves. We call them IHEU. And in some countries, due to the success of prevention of mother-to-child transmission efforts, these children make up to 30% or more of the population's children. And the reason this is important is that these infants have been shown to have higher morbidity and mortality regardless of the setting. This is somewhat abrogated potentially by breastfeeding, but not completely. And it's mostly due to susceptibility to infectious diseases thought to be due to altered immunity. So these infants, HIV-exposed infants, really need to be protected against infections with vaccination. And because breastfeeding is recommended by the WHO for most women with HIV, these exposed infants are a key population for HIV vaccines. But they have altered immunity, as I mentioned, including an immune activation and altered T cell responses to vaccination. And since women with HIV have altered vaginal and gut microbiota, it stands to reason that we and others have found that these infants exposed to HIV have altered early life gut microbiota, including lower relative abundance of bifidobacteria. So we hypothesize that this altered gut microbiota in infants exposed to HIV contributes to their poor vaccine responses. To test this hypothesis, we are utilizing our longitudinal cohort of HIV-exposed and unexposed infants that recruit in Khayelitsha, Cape Town. And for the purposes of this analysis, all the infants were term vaginal deliveries. The exposed infants had a negative HIV DNA PCR, and all of them were at least initially breastfed. We collect stool, blood, and other samples at multiple time points through the first year of life in these infants. And just to remind you, BCG is given within the first couple of days of life. To measure BCG-specific immunity, we take blood and incubate it with BCG media alone or a positive control within 1 hour of phlebotomy. We do a short-term incubation before freezing, and then at some later time point, we defrost the cells and stain them with a multi-parameter flow cytometry panel, including the Th1 cytokines, interferon gamma, TNF alpha, and IL-2. And you can see that when we look at the net total percentage of CD4 T cells making any Th1 cytokine, that there's quite a variability in response. And these responses correspond very, very, very well with COMPASS polyfunctionality score. And for the purposes of this analysis, we binarize these responses into high and low responders around the median net Th1 cytokine response. We looked at the gut microbiota in the first couple of days of life around the time that BCG was given in relation to the week 7 BCG response, which is the peak response we find in infants. And there were no significant differences in alpha and beta diversity between infants that were high and low responders. In summary, I think I've shown that certain gut microbes in early life are important in immune ontogeny, that specific gut microbes could influence CD4 T cell responses to vaccines. Bifidobacterium longum subspecies infantis has the potential as a probiotic to improve Th1 responses in neonates, such as important for TB or HIV vaccines. And bifidobacterial metabolites could be further explored as prebiotics or adjuvants for vaccines. And our next step is to perform a randomized controlled trial of B. infantis versus placebo to improve immunity in infants exposed to HIV. And with that, I'd like to thank Dr. Donald Nyangahu, who performed the majority of the mouse experiments and who is currently looking for faculty positions. The rest of my lab in Seattle at the University of Washington, Seattle Children's, the University of Cape Town lab and the clinic for performing the human recruitment and immunity. And my UCT collaborators, Clive Gray and Jonathan Blackburn, the Erdal lab for teaching us how to do the tetramer technology, participants in our studies, and of course, the funders of this R01. Thank you very much.